Let us begin with the first type of chromosomal theory that was proposed in order to understand the sex determination. This theory is known as Theory of Heterogamesis. This theory was first proposed by a scientist named Corinth in 1906. So, according to this theory, one sex produces two types of gametes. That determines the sex. And this determination of sex takes place during fertilization. This type of theory is basically of two types. First, XX XY type, and second, XX XO type. Thus, these are the two types of that are proposed on the basis of theory of heterogamesis. Now let us begin with the first type, that is XY type. In this type, mainly two different patterns of sex determination are observed. The first is XX female and XY male type, and the second one is the opposite, that is XX male and XY female type. So let us first of all begin with the first, that is XX female and XY male type. This type of pattern of sex determination is generally observed in insects like Drosophila and in other organisms like humans. So let us first of all understand the sex determination of this type that takes place in Drosophila. This sex type of determination in Drosophila was first observed by a scientist named Morgan. According to him, in Drosophila there are mainly 4 pairs of chromosomes. Out of which 3 are autosome and 1 is sex chromosome. So, the 3 autosomes that are present occurs in the form of 3AA, while the single pair of sex chromosome that occurs is either XX or XY. XX occurs in females, while XY occurs in males. So, in females, the sexual arrangement of chromosome is in the form of 3AA plus XX, and in males it is in the form of 3AA plus XY. So, on this basis it was proved that the female produces only one type of ova, that is 3A plus X, while the males produce two types of sperm, that is 3A plus X and 3A plus Y. So, it is on this basis that crossing or mating between which type of sperm and ovum occurs that yields the development of an individual after fertilization. For example, if the fertilization between a male and female Drosophila occurs in the form of the sperm is 3A plus X, while the ova is 3A plus X. That is very obvious that ova occurs only in this form, while sperm. So, the resultant can be only 3AA plus XX, that is, which produces females. And the second type of mating that involves the second type of sperm, that is 3A plus Y, into ova, that is 3A plus X, forms 3AA plus XY, that is the male Drosophila. So it depends on the male parent that which type of sperm is involved in mating with the female ova and results into the formation of an individual. Similar pattern of sex determination is also observed in many other plants and animals. This figure depicts the sex determination in Drosophila, where the crossing between female and male parent is done. And based on the genotype, the F1 generation is obtained, which further undergoing multiplication gives F2 generation, where either XX produces female or XY produces male, depending on the type of sperm that becomes involved during the process of fertilization. Similar type of method is also observed in humans. So let us understand about the sex determination in humans. Like Drosophila, humans also exhibit the XX female or the XY male method for determining the sex. But unlike Drosophila, which contains 4 pairs of chromosomes, in humans there occurs 23 pairs of chromosomes, in which 22 pairs are autosomes while the 23rd or the last pair, that is one pair, is sex chromosome, which is also called allosomes. Similarly, like Drosophila, here the female chromosome contains only one type of chromosome, that is X, while male contains Y. That is, in female 2X occurs while in male XY. Thus, on this basis it can be said that each egg contains 22 autosomes and one sex chromosome, that is 22A plus X, while the sperm contains two different types, that is 22A plus X and 22A plus Y. So, whether the child will be boy or girl depends on the kind of sperm that becomes involved with the female ova during the process of fertilization. This figure depicts the cross-fertilization between male and female, where depending on the sperm type, the mating between two occurs and the resultant individual can be either boy or girl depending on the type of sperm that gets involved in the process of fertilization. Following it comes the second type of method, that is XY female and XX male method. As it can be seen that this method is entirely opposite to the previous one. In this type of sex determination, the female is usually heterozygous in nature, that is contains two different types of chromosome, that is X and Y, while male contains homozygous type of chromosome, that is of the same type, which is only X. Thus, in the first one, that is XX chromosomes are present in males, such a type is involved in organisms like bugs, and grasshoppers. Since such a type of homozygous or heterozygous nature of male or female depends and is responsible for the sex determination of an individual. Other example which best suits to this method is in birds. As said earlier, in birds, the ZW chromosome represents female, while 2Z chromosome represents the male. This is entirely opposite to the previous one, where the males produce only one type of sperm while the females produce two types of ova. Here, in case of birds, the X and Y chromosome are represented in the form of Z and W, and accordingly, on the basis of fertilization process, the resultant bird would be a male or female, depends on the type of ova that is involved in fertilization.